Hello. Good afternoon.
Hi.
Hope everybody's well. We still a couple minutes till 4 o'clock, so.
Yeah, I think I was the only one here.
Welcome. Happy Wednesday. Yeah, if you would, go ahead and type your first name or your nickname into chat for me. One of my dogs has learned that when I sit down at the table now, that means I'm going to be busy for a while. And so as soon as I sit down, she comes over and tries to get in my lap now. And she weighs about 50 pounds, maybe. So not exactly a lap dog, but she has definitely decided that it's not okay that I don't pay attention to her while I'm teaching. I'm genuinely concerned about my dogs for when I go back to work and I'm teaching in person. I'm genuinely concerned about what's going to happen to them because they have gotten so accustomed to me being home all the time now. I think they're going to have terrible anxiety when I when I finally do go back to teaching in person. Welcome in. Go ahead and type your name into the chat for me if you haven't. Your name or your nickname, either one is fine. Good to see everybody. I hope everybody's doing well. It's a good time just to remind everybody that if for some reason, heaven forbid it, but if for some reason, you get ill, especially if you are diagnosed with COVID or somebody in your family is diagnosed with COVID, again, heaven forbid, please let me know. I have a couple of students from a different school who are sick with it now, and I want to know so I can work with you and we can make things less difficult for you. So if you're comfortable, let me know, and I will be sure to try to help give you time that you need to finish your work and stay in the course and so on. All right, very good. Looks like just about everybody's here. I think we're missing one still, but doing well. Today in lab, we're going to continue talking about how we can control the growth of microorganisms. But today, instead of talking about a physical method like heat or ultraviolet or pressure, we're going to talk about chemical methods. And by chemical, I mean the kind of chemicals that we use to disinfect, essentially. So that's our topic for today. Before I get started on that, though, does anybody have any questions about anything? Any questions about any of the lab material or the lecture material? Anything at all? Feel free to type it into chat, or if you'd rather, you can unmute yourself and speak out. Feel free to do that. Okay. I want to just pull up our Canvas page for a moment so we can take a look at our schedule. Again, just as a reminder for us, it's always good to remind ourselves what's coming up. So I'm on our modules page. We're obviously right in the middle of this week here. It's March 3rd. So the topics this week, again, are about how we do things to control microbes, to control their growth in our environments and also in our bodies. So we were talking about ultraviolet the last time we were together, and today we're going to be talking about chemicals. Now, I did post a document. One of you had contacted me and asked if I could post this summary table that I mentioned in the lecture about chemical disinfectants. I have posted that for you. If you find that helpful while you're studying or taking notes or anything, this is available to you. And of course, the lab slides that we'll use today are here as well. I just want to point again to next week. It's a very unusual week for us. We don't have any lectures to cover next week, no lecture quizzes, and we have no lab material for next week. So if I click here on our to-do list, it's really just instructions for the 2 exams you'll be taking next week. You have the second lab practical exam on Monday, and then you have your 3rd lecture exam on Wednesday. So it's only going to be those things next week. There's no other work for you to complete. That'll be plenty though, right? That'll be plenty of work for next week.
Can we do those? Can we do those early?
What do you mean by early?
I'm starting a new job on Monday, and I'm not sure when my hours are going to be.
Oh,
You know, hoping I could do them Sunday.
Well, send me an email. I can set it up for you separately from other people. So yeah, absolutely. Send me an email message and we'll take care of that for you.
All right, thank you.
You're very welcome. And congratulations on your new job. Okay, and I do remember that you had also, some of you had requested that slides be posted so that if you wanted to get ahead a little bit during the spring break week, you could do that. Those are scheduled to come up, so you'll have some extra lecture material. If, again, you have the time and you're able to put that time in, dedicate it to that, that will be available to you during that spring break week. So, all right. Any questions? What I'm going to do is go ahead and pull our slides up and we can get started. So you should be seeing on your screen a slide that says controlling microbial growth with chemicals. And our objectives for this exercise are to examine the 4 most common families of disinfectant chemicals, to compare the relative effectiveness of those chemicals, and also to compare the limitations of those chemicals. Now, we'll also need to learn a little bit of new vocabulary because there are a lot of terms associated with these chemicals that can kill and control the growth of microbes. So we'll start off by doing that. This word disinfectant or disinfect, this word actually means something very specific. You may have heard other terms like sanitize or antiseptic. All of these things are related to these types of chemicals, but they mean different things. They have different strict definitions. When we talk about a disinfectant, we're talking about a chemical that is designed to, I shouldn't say chemical, we're talking about something that is designed to eliminate microbes, specifically pathogens, and specifically from inanimate objects and surfaces. So we are talking here about things that are not alive. You disinfect things that are not alive. So you disinfect your tabletop, you disinfect your sink, you can disinfect, all different kinds of objects and surfaces. If you're talking about living things, you're talking about antiseptics. So if I am applying a chemical, for example, to a wound, because I'm trying to kill any microbes that might be in and around that wound, I'm talking about an antiseptic. So the kinds of things that we use as disinfectants, we would not apply to living tissue, at least in some cases. There are some chemicals that can do double duty. One of the most obvious is alcohol. We use alcohol on living tissue, but we use it fairly sparingly because we know that while it is good at killing microbes quickly, it's also quite drying to the skin. So again, in different circumstances, alcohol is appropriate, but in other circumstances, we don't use it. And we could also use alcohol as a disinfectant. We've talked about that. Remember, the 60 to 90% solution makes a really nice disinfectant. When you hear terms other than antiseptic or disinfectant, you're talking about some kind of a level of protection against microbes that is less than what you would get from a disinfectant or an antiseptic. So anytime you see this term sanitize or sanitizer, that's a much lower level of protection than a disinfectant or an antiseptic. When you go to the grocery store or to other stores that sell chemicals like this, cleaning chemicals, next time you go, look at the labels, and you'll notice that a few of them use this term sanitize. Again, the chemical is intended to be used to remove microbes from our environments, from our surfaces, but it's not powerful enough to eliminate the common pathogens that we encounter. So they can't call it a disinfectant. That's where that word sanitize comes in, or sanitizer comes in. Now there's a couple of other terms on this slide that we need to know. One of these is bactericidal, and the other one is bacteriostatic. Two different terms that describe two different mechanisms. And these are applied not only to chemicals that control microbial growth, but also to antimicrobials and antifungals and antibacterials and so on. When you hear this word bactericidal, this translates as bacterial death. So these are compounds, chemicals, or drugs that kill bacteria. The bacterial cells will actually be destroyed. They'll be killed. In comparison, something that is called bacteriostatic is something that stops the growth of bacteria. So it's not that the cell is going to die, but the cell will not be able to divide and make more cells. So those are two different categories of effectiveness against bacteria. Now, there are lots and lots of chemicals on the market. And again, you can walk into any grocery store, you can walk into any office supply store, you can go into CVS or Walgreens. There are online retailers who sell more commercial products rather than residential. There's lots of products. And the truth is, there is no single chemical that we can rely on in all situations for all things. We have to match whatever we're using to a couple of things. Number one, what are we trying to control? What microbes do we think we might be trying to control in that situation? And number two, what is the environment like that we're trying to apply the chemical to? It's very different to apply, for example, chemicals to human skin than it is to apply something down your throat and into your stomach, right? So the kinds of things that I might apply to the outside of my body to control the growth of microbes are going to be very different from anything that I would swallow to treat the inside of my body. And similarly, if I'm trying to disinfect stainless steel, That's a very different situation than if I'm trying to disinfect, say, fabric or some kind of linens. You have to take into account not only what kind of microbes you think you might be encountering, but also what kind of materials that you're trying to apply this chemical to. So these are things like, when it comes to the environment, the material itself, of course, the pH of the environment. Is it a neutral pH? Is it just some plain surface? Or am I trying to apply it to something that has a low pH or a higher pH? How soluble is this chemical? That's going to matter if I'm applying it to something that stays wet in the environment. Is that chemical going to be able to dissolve, or is it going to stay as a solid? All of those kinds of things matter. Toxicity, of course, is important. And toxicity can change depending upon which tissues we might be applying antiseptics to. All of these things matter. One that you may not think about is this last one. When we're disinfecting inanimate things, the amount of organic material that might be present on that thing really matters. Think of organic material, think of, old food. If you're trying to disinfect plates and utensils, it matters if there's food crusted onto those plates, right? You need to clean things before you disinfect them. And the same is true for things like clothing, any kind of linen material you might be trying to disinfect. If it's got a lot of organic material on it, that organic material is going to interfere with the action of the chemical. Sometimes organic materials actually inactivate chemicals. So a very dirty garment, for example, should be cleaned before you try to disinfect it. Organic matter really can get in the way. Something else to remember, just generally speaking, a chemical can be effective against one type of pathogen and be completely ineffective against another. So you have to know your chemicals before you use them in an attempt to disinfect or to use as an antiseptic. Now, I want to spend a few minutes talking about a particular quantitative measure of controlling bacterial growth. And this is referred to as decimal reduction time, or DRT. In some source materials, you will hear it called the D value, but I almost never see it referred to as that. DRT is the most common way that this quantity is referenced in source material. What DRT is the time in minutes that's required to kill 90% of some sample of microbes in some circumstance. So, for example, if I'm applying alcohol to a stainless steel table, in order to calculate this DRT, I would have to know what's growing on the table, what's growing on this stainless steel surface, how many cells are there to start with. Then I apply the chemical according to the directions, and I should see a reduction of 90% across, a certain amount of time. That's going to be my DRT. When chemicals are evaluated to determine whether or not they can be called disinfectants or antiseptics, this DRT quantity has to be determined. So these chemicals are sent to the laboratory and they're evaluated for their effectiveness. If it takes, 2 hours of contact time to reduce the microbial load on a surface by 90%, you need to put that on the bottle. You need to tell the user Well, you can use this as a disinfectant, but you have to leave it on the surface for 2 hours. Now, not many people would want to buy that disinfectant. It's just not very practical. But if the DRT comes in at 2 minutes or, 30 seconds, that's a different story. But that's what DRT is. It's the literal amount of time that it takes a chemical to reduce a certain number of microbial cells by 90%. These kind of quantities, these DRT quantities are published, and you can look this stuff up for individual types of chemicals. Now, when we're evaluating these chemicals and we're calculating DRT, we also have to do this type of testing several times for each chemical because, as I said a minute ago, the efficacy or the effectiveness of a chemical can vary for different kinds of microbes. I mean, if you think about just the bacteria, the gram-positive versus gram-negative bacteria, they have a different anatomy, right? The gram-positive cells have a thick layer of peptidoglycan around them. The gram-negative cells have a thin layer of peptidoglycan and then a second membrane, an outer membrane. So it's not surprising that some chemicals are more effective against some organisms than others. So this slide, it's very hard to read. I'll try to enlarge it a little bit here for us. But this is a table that comes out of a textbook. And it looks at several different types of chemicals that are effective at controlling the growth of microbes. It looks at the pH of the chemical. It looks at the types of bacteria that they're effective at, what you need, what concentration you need to mix it up to in order for it to act as a disinfectant. And then you get these different values. So you can see this table records it as D value. So this is the DRT. This is the time in minutes that it takes these chemicals to destroy 90% of the microbes on a certain surface or in a certain situation. 3.8 minutes for hydrogen peroxide to destroy Pseudomonas aeruginosa, 90% of them gone. 5.6 minutes for it to kill Bacillus subtilis. 8.7 minutes for Staph aureus. Same chemical, right? Same chemical, but it needs to be at different concentrations, and it takes longer periods of time or shorter periods of time, depending on what organism you're trying to kill. Again, this is what I mean when I say it's really important for us as people who might be in charge of disinfecting things at our work. It's really important for us to know our chemicals because you can very easily use these chemicals incorrectly, and then they're not going to do the job you need them to do. So there are lots of these kinds of tables available to us online and in textbooks and so on. Now, in addition to the DRT statistic that you can look up for chemicals, there are other tests that get done in order to examine how effective a particular chemical is as a disinfectant. If you look at this table that I just showed you, you'll see for each of these different chemicals, they're applying them to 3 different organisms. These are the organisms that are usually used when we are evaluating chemicals. And that's because they provide a wide range of types of bacteria. So Staph aureus, Bacillus subtilis, and Pseudomonas aeruginosa. Both the Staph and the Bacillus are human pathogens, can be human pathogens, some strains, but one of them's gram-positive and one's gram-negative. And the Pseudomonas is an environmental organism. It can cause infection in humans, but typically in people who are immune compromised a little bit. So you get a wide view of bacteria when you look at those three organisms. So when these chemicals get tested in the lab for their effectiveness, the test often checks the chemical against those three organisms. You see it again and again. So on this next slide here, the American Official Analytical Chemists, that's a group called the AOAC, do a particular evaluation called the use dilution test. Now, what they're doing is they're taking those three test organisms again that I mentioned, including, you'll see there's another one listed here, Salmonella, Enterica, Cholera Suisse. How's that for a name? So the genus name is Salmonella, the species is Enterica, and the strain is Cholera Suisse. So again, Staph aureus is here. Staph aureus is a human pathogen. It's a gram-positive coccus. The Pseudomonas is here because, again, it's an environmental opportunistic pathogen. And then the Salmonella is a gram-negative organism that tends to live in the intestine. So we have a nice range of bacteria here to test these chemicals against. What the procedure involves for use dilution is you take these little metal cylinders, they're made out of steel. I'll show you these in just a minute. They refer to them as carriers. And you drop the cylinder into a broth culture of each one of these organisms. So one cylinder per broth culture. Then you take it out of the broth culture and you set it down and let it dry. So essentially you're coating this stainless steel cylinder in some kind of microbial cells. Then you set up a little container that has this test disinfectant in it, and you place the metal, the stainless steel cylinder into the container, and you leave it for the recommended time. And then you take it back out of that container and put it back into broth. And you see what will grow. You can quantify after that test how many of the cells survived. So I take that little piece of metal, I coat it basically in bacterial cells, and I let it dry. So I've got a piece of metal covered in bacteria. And then I take that tube and I submerge it into the chemical for however long the manufacturer says it needs to stay in contact. Then I take it back out and I put it into some fresh nutrient broth, and I see if any cells survived. And if they did, I grow them up. I can quantify how many cells were killed and how many cells survived by doing a little mathematics. So this test, it's common, it's done commonly enough that I want us to be familiar with it. But in my experience, DRT values are done more often than the use dilution test. Now, the thing about use dilution testing is that it only works for chemicals that are listed as bactericidal. So this would not work against a chemical that was listed as bacteriostatic. And the reason it wouldn't work is because it's not intended to kill anything, right? If it's bacteriostatic, all the manufacturer is saying is that it will stop bacteria from dividing. It's not going to kill them outright. So the only use for this use dilution test is when you're testing chemicals that are bactericidal, that are designed to kill. So on this slide, you're looking at, these are Petri dishes, but they're glass Petri dishes. These are reusable. And what's going on in here is these little cylinders, they have been dipped into culture and they're now sitting inside the sterile petri dish drying. So these cylinders have had cells applied to them. They're going to dry. And once they're dry, they'll be dipped into whatever chemical is being tested. Now, there are other tests that people do when they're evaluating disinfectant chemicals. You can modify essentially the DRT or the use dilution test, but it's kind of done more informally. So, for example, it's possible to take a broth culture, so a tube that has bacteria growing in it, and just expose it to a disinfectant. You can put certain disinfectants, right into the broth culture, let it sit for a certain amount of time, and then pull out some cells and see if any have survived. It's a little bit difficult to do this well, like do it with any accuracy, because you have to make sure that what's killing the bacteria is the chemical and not some physical effect of adding the disinfectant. So you have to understand a little chemistry if you're going to do a test that way. You have to understand the type the chemical is, the type of molecule the chemical is, and whether it's having a physical effect or whether it's having an actual chemical effect. And that can be really tricky. So that's what I mean when I say these are more informal tests. The reason that someone might want to do a test this way is because it allows all of the cells to be uniformly exposed. If I add a small volume of disinfectant into a broth tube, I can mix that tube up and make sure that all of the cells in that liquid culture are getting exposed uniformly. It's a little harder to ensure that when you do something like the use dilution test. If I'm going to place the cells in on this metal cylinder, maybe the cells on the inside of the cylinder are a little more protected than the ones on the outside are. That's why some people like to put the disinfectant directly into the broth. So there are a couple of very formal, very quantitative tests available to us. And by quantitative, I mean we're actually going to calculate numerical values, like the DRT determines the time necessary for that 90% reduction. But then there are other tests that are more qualitative. We're just saying whether or not cells can be killed. And again, it all depends upon the needs. What are you trying to do with this testing? Are you trying to figure out if a chemical is going to work for you in your situation? Maybe you own a manufacturing plant and you just want to know if I choose bleach as my disinfectant, is it going to be effective against the kind of organisms that I usually run into? Or would I be better using alcohol? So that's kind of informal testing. But when a manufacturer wants to label a chemical as a disinfectant and wants to sell it as a disinfectant, then we need more standardized, more quantitative testing, like the DRT or the use dilution test. So it's good to know, for example, that these kind of tests take place because, when we go out and we purchase these materials, particularly as individuals, as homeowners, we want to make sure that what we're buying is effective. We want to know that it's going to do what it claims to do. You really saw this this year during the pandemic. You really saw people being a lot more aware of what chemicals they were using specifically to protect themselves against the SARS-CoV-2 virus. you saw a lot of people asking questions. Well, if I use alcohol, do I have to let it dry or can I wipe it off? And if I'm going to use bleach, what strength bleach do I need? And a lot of people were asking some really good questions this year. And I think people started to read labels more. I think I mentioned to you when we were talking about maybe it wasn't this class, but I was recently talking with students about the pandemic, and we were talking about the things that were not available a year ago. If you remember, there was a terrible run on toilet paper. And for like a month or two, we couldn't find toilet paper anywhere. And it was very difficult and stressful. And all the way throughout the summer, and maybe even now, I don't really know. There have been reports that you couldn't buy Lysol cleaning wipes. Lysol disinfectant wipes. I know one of my siblings loves these wipes and she uses them all over her house. And she was very upset that she couldn't buy them over the past several months because of the pandemic and they were low in supply. And one of the things that I noticed in my local grocery stores was that they were trying to replace the Lysol disinfectant wipes with other brands. So they were telling people, well, we don't have these Lysol wipes, but we have the Market Basket wipes or we have the Shaw's wipes. And if you looked at the label on some of these, it was very clear that it was not antiviral. It was antibacterial, but not antiviral. I think a lot of people have started to realize now that chemicals behave differently and you have to read the label if you want to make sure that you're getting a level of control over microbes that you need. You have to read the label. You have to know what chemical you're using. You have to know how to use it, how long to leave it on the surface, whether it should dry, whether you should wipe it off. And you need to know what it's effective against. And if you're worried about viruses, like we all are right now, you need to ensure that you're buying something that's effective against viruses. Did any of you have any difficulty finding any sort of disinfectant products this year, or were you able to find the things that you needed? I know for myself, I kind of got lucky because I found wipes that had alcohol on them. I prefer alcohol to bleach or anything like that because I tend to ruin my clothes with bleach. And I found that a local store was selling packets of alcohol wipes, and I found those very useful. Throw a package in the car, and I could clean my hands off with alcohol after I touched things, Did anybody have any trouble? Did you guys have trouble buying toilet paper back last spring? I couldn't believe it. It was the strangest thing I've ever encountered. Not to be able to find something as simple as toilet paper, but there you have it. All right, let's jump back in. So the next thing we're going to talk about, and really for the rest of the lab today, we're going to talk about 4 families of chemical disinfectants. You could think of these as being classes of disinfectants. So this is not one chemical per group. This is several chemicals per group. These are the 4 most commonly used chemical classes that people use as disinfectants, and in some cases, antiseptics. The first group are the alcohols. Then there are the chlorinated compounds, the Iota Fours, and the Quaternary ammonium compounds. So let's start with the alcohols. Now, there are lots of alcohols that are used as disinfectants. You can use ethanol. You can use the kind of alcohol that that you might find in alcoholic beverages. You can also use alcohols that you would not want to consume because they would injure you, things like isopropanol or methanol or other types of alcohol. Alcohols are a big family of chemicals. Now, they also happen to make good disinfectants if you use them correctly. So when it comes to their effectiveness against bacteria, they are rapidly bactericidal. Again, at a 60 to 90% solution. So by rapidly, I mean within minutes. They need to be at this diluted level in order to have that level of effectiveness. If you apply alcohol undiluted, it's less effective. It turns out that the water that we add into alcohol to dilute it actually aids in the effect of the alcohol and makes it kill these bacterial cells quicker. So people are always surprised when I tell them that they should dilute their alcohol if they're going to use it as a disinfectant. We want to think that the purer a chemical is, the better it will be. But the truth is, when it comes to alcohol and bacteria, the diluted form is better. Now, what kind of bacteria and other microbes is alcohol effective against? We're going to use this same list as we compare all of these classes today. We're going to ask, is it effective against bacteria? Is it effective against the organism that causes tuberculosis? That organism has a genus name, Mycobacterium. Remember, this is an organism that has a very unique characteristic. This is an organism that you can't gram stain. And if you remember, that's because it secretes this waxy material that covers the cells and covers the colonies in the laboratory. So you end up with what we call gram indeterminate. You can't gram stain them. Even if you have a healthy young culture, they just won't pick up the stains because the stain can't get through the wax. So Mycobacterium gets its own sort of special place when we're evaluating chemicals because of that waxy material. It is just a harder organism to kill. So sometimes when we're evaluating chemicals, we think of Mycobacterium separate from the other bacteria. All right, let's jump back in. So we said it's rapidly bactericidal if you use it at the right dilution. It is effective against Mycobacterium. That's great. It's effective against fungi. It will kill fungi. Yes. It will kill viruses. Yes. But here's an important thing to note. Alcohol is not effective against spores. Remember, spores are really hard to destroy. A spore is, by its nature, very, very resistant to anything that might try to destroy it. So we have to know what will and will not destroy spores if we are trying to control spores. If we have a feeling in our particular situation that spores might be an issue. Now, what exactly does alcohol do when it contacts a bacterial cell or a fungal cell or a viral particle? Well, it has one primary effect, and that is it denatures protein. Now, this word denature means essentially it unfolds protein. Remember, proteins are just long, long chains of amino acids. And after the chains are assembled in cells, they get folded up into these very complex three-dimensional shapes. And as we learned in lecture, it's the shape of a protein that determines its function. So when alcohol denatures protein, when it unfolds the protein, it interferes with the function that it does for the cell. And cells cannot survive without their functional proteins. Now, alcohol is a great disinfectant. You've heard me say I really like it as a disinfectant, but it does have some limitations that we need to know about. For example, there are some materials you don't want to apply alcohol to all the time because over time, the alcohol will destroy them. For example, rubber, plastic, and lenses. You don't want to apply alcohol over the long term to these kinds of chemicals because the alcohol will degrade the chemical over time. The rubber and the plastic, you can actually see it happening. It's a little harder to tell when you're damaging lenses. But, and by the way, I'm talking about, when I use that term lens, I'm usually talking about glass lenses, like the kind you would use on a microscope. The alcohol over time just degrades the surface of the lens, and it will break down rubber, and it will break down certain plastics over time. So again, you have to know, what am I trying to disinfect, and what is it made out of, and what chemical would be the best, both to disinfect it, but also to not destroy it over time. So damage to materials is certainly a limitation of alcohol. But here's another one. Alcohol, pure alcohol that you would buy in a large container like this, is very expensive. It's much more expensive than some other disinfectants. When we go into the drugstore, maybe to buy a container of isopropanol or something, like rubbing alcohol, we don't think of it as being super expensive, but when you have to buy gallons of alcohol to use in your hospital or to use in your laboratory or the doctor's office or anything like that, it can get very expensive very quickly. Most pure alcohols are coming in now at about $100 a gallon. So again, depending on how much you need, how much your facility goes through, it may be good, or a good economic choice for you. But a lot of places it's just too expensive. So they're not using alcohol routinely for all of their disinfecting needs, I guess I would say. So Debbie's got a comment here in the chat. I did want to talk about. Debbie's asking about the future of antibiotics as, again, a way to control microbes. Now, when we're talking about antimicrobials and particularly antibacterials, I'm sure you're all aware that we have a real emerging problem with resistance. And in recent years, I'm going to say maybe the last 20 years, there's been a real push in medicine, both human medicine and veterinary medicine on my side of things. There's been a real push to back off of prescribing antibiotics quite as freely as we always have. When I was a girl, I can recall going to the doctor's office and, my mom telling the doctor she has a sore throat or she has an earache. And I remember the doctor always gave my mother antibiotic to give me. The thinking was, well, it may be bacterial, it may not, it might be viral, but let's give her the antibiotic just in case it's bacterial. That was kind of the thinking back then. And I can tell you, when I first became a veterinarian, we had a very similar sort of approach. We liked to sort of cover ourselves just in case there was an infection going on. What the push now is in medicine is, don't prescribe it unless you know it's a bacterial infection. And use the antibiotic for as short an amount of time as possible. That's the other thing. When I was growing up, all antibiotics were given for 10 days. It didn't matter what kind of antibiotic you were prescribed. You took that antibiotic for 10 days. Now what we're learning is we can get away sometimes with 5 days or 3 days. There are even a couple of the newest types of antibiotics that only require 1 or 2 doses. So we're really trying hard now. Number one, not to prescribe when we are not sure there's a bacterial infection. And number two, to only prescribe for as long a period of time as necessary to get the infection under control. Now, Debbie's also asking about cephalosporins. In the antibiotic lecture that you guys will be viewing if you haven't already, you know the cephalosporins are basically, they're a spread out group over several generations, as we call them. The cephalosporins sort of were born out of the penicillin type drugs. And the cephalosporins have lots of advantages over the penicillins, but they are related to them. And as I said, there are several generations of them. In other words, we're always coming up with new cephalosporins because we can tinker with them in the lab. Cephalosporins are a really important group of antibiotics right now, because the truth is some of our other penicillin type drugs just have a whole lot of resistance to them. The resistance problem is real. We are seeing more and more resistance. Now, here's the thing that some people don't understand, and it's important to know. A lot of people think that overuse of antibiotics has brought on resistance, that it's all because we were prescribing antibiotics when we shouldn't have been. That did push resistance a little bit, but here's the scary thing. Any use of antibiotics, including the appropriate use of antibiotics, will push towards resistance over time. Anytime you use antibiotics for a bacterial infection appropriately, what you're doing is you're selecting out the resistant organisms. You're allowing them to thrive. So over time, and again, it does take a long time, we start to see more and more and more resistant bacteria. So even if we use antibiotics absolutely perfectly, we only ever give them to people who we know have bacterial infections, and we only give them for exactly how long they need to be given. Even if we do everything perfectly, you would still see resistance. It's a problem. It's a big problem. There's a lot of research going on right now to try to find new antibiotics. We're just in a race now to try to stay ahead of the bacteria and keep trying to find new drugs that can control their growth. Oh, yeah. Oh, there's a great question from Debbie about growth promotants. Of course, I'm a veterinarian, so this is an area that's near and dear to my heart. You may have heard, I'm sure you've heard, because if you've ever bought meat in a grocery store, you've seen the label, right? That says no antibiotics. This is a huge thing now in the marketplace. We as veterinarians have been putting antibiotics into the food of livestock for decades, close to 100 years now, all right? Here's why. We started feeding tetracycline specifically to cattle, sort of as a result of an accidental finding. Somebody discovered 100 years ago that cows that were given tetracycline because they had an infection, they also happen to grow faster. So they started doing experiments feeding tetracycline to cattle. And what they discovered was if you give cattle a low level of tetracycline in their food, they will grow faster. They'll get bigger and meatier faster. It's important to know that they don't give them enough tetracycline to be an antibiotic. They give them a very low dose. In other words, this is an effect of tetracycline that has nothing to do with killing bacteria. It's an effect we don't really understand, but we know it happens. It happens in cattle and it happens in other what we call food animals. So for decades, we have put tetracycline into animal feeds to help the animals grow. But with the emergence of more and more and more resistant bacteria, that has fallen completely out of favor. What a lot of folks don't realize is a law was passed several years ago, it might be a decade now. A federal law was passed that prohibits putting antibiotics into animal feed. You can't do it anymore. So it's not happening anymore. But the people who make meat products still pretend that their meat is better than another person's meat because there's no antibiotics in it. Even when we fed tetracycline to animals, the other thing that you should know is the farmers are not allowed to sell the animals for slaughter without taking away the antibiotic for a couple of months. In other words, when an animal goes to slaughter, they can't have any antibiotic in their system. It's not allowed. It's never been allowed. So by the time the animal goes to slaughter, it has had a couple months to get the antibiotic out of its system. They stop feeding it about 2 months before the animal goes to slaughter. So the hard truth is that antibiotics in our meat has never been a problem. It really hasn't, not in this country anyway. And nowadays, they're not even allowed to put antibiotics in feed. So it's certainly not a problem right now. But the market has realized that if you put no antibiotics on the package, you can charge more for the meat. People will pay more. They'll pay a premium for that. So it's really deceptive now to put that on a package of meat. There is no antibiotics in any meat that's sold in the United States. So it's deceptive labeling. But everybody does it. Everybody does it. If you walk in, if you look at the Purdue meat, if you look at the Tyson meat, if you look at the ground beef, if you look at it doesn't matter. It's got it on the label. Yeah, it's a really interesting and really deceptive practice that they're doing right now. But hopefully it makes you feel better to know, because it makes me feel better, that nobody is allowed to put antibiotics into feed anymore. That stopped. That stopped years ago. Great questions. I'm always here for these animal related questions. All right. I'm going to jump back in now. This slide that you're looking at here, this is just an artist's diagram of what alcohol is doing to protein. I'm a visual learner, so I like to see images of what's happening inside of cells. This is what we draw proteins like. You may have seen images like this in textbooks. What this represents, this green ribbon, represents a long string of amino acids, and they're all bound one to the other. Most proteins have hundreds to thousands of individual amino acids in them. When that string gets manufactured in the cell then folds that string up and you can see there's all kinds of twists and turns in this, that creates a three-dimensional molecule. And every single protein has a unique shape because every protein has a unique function to perform in the living organism. When you apply alcohol to this kind of structure, this happens. So you're not taking the amino acids apart. You're not down to that level of destruction, but you are denaturing it. You're unfolding the proteins. And bacterial cells, they can't survive that. They're going to die because their proteins won't be able to do the work that they normally do if they get unfolded by the alcohol. On this slide, we're looking at a diagram of a spore. And you can see the name that this structure is given is an exosporium. Oh, sorry. I guess they're labeling just the outside layer of this, the exosporium. You can see that there's a layer labeled cortex and core. Don't worry about these terms. I only put this diagram in here because I want to drive home that spores are built to resist destruction. Remember, this thing came from a living cell. The cell got rid of all of it, the parts of it that were vulnerable to destruction when it became a spore. In the very center of this structure, there's the genetic material for that organism. And then there are layer upon layer upon layer of protective material around that. So these are very resistant to destruction. Now, the next class of chemicals are the chlorinated compounds. The most common chlorinated compound used as a disinfectant is called hypochlorite. And this is simply household bleach. So hypochlorite is the most common of many chlorinated compounds that will kill bacteria. Plain old household bleach, the same kind that you buy in the grocery store. Now, just like alcohol, it is rapidly bactericidal, but you have to dilute it. And you dilute bleach way down. You only need a 10% solution to kill bacteria. When we look at the scope of activity, again, against the same organisms, it is effective against the bacterium that causes tuberculosis, but you have to leave it in contact with the surface for several minutes. So that's an important thing to know. If you're disinfecting a facility where you know patients have TB, bleach is not going to be your choice as your disinfectant. Bleach is effective against fungal cells. It's effective against viruses, but you've got to leave it for several minutes. You've got to leave the bleach in contact with the surface for several minutes. So again, bleach turns out not to be the best antiviral type of chemical. Alcohol is a much better choice when it comes to viral particles. Now, here's something that I find really fascinating. When it comes to the mechanism of action, in other words, what is it that bleach is doing that kills bacteria and kills fungi and destroys viruses? The answer is we don't know. At least we don't know completely. You would think that surely we could figure that out, but the truth is the mechanism is still not completely understood. One thing we do know is that when you take bleach and you put it into water, it forms an acid. And that acid, it's a weak acid, but that acid is an oxidizer. And when I use that term, what I'm saying is it can steal electrons away from other molecules. So bacterial cells that are exposed to a diluted bleach solution are going to suffer because their molecules are breaking down as electrons are being stolen out of the molecules. It's kind of fascinating that we don't really understand how this works. Oh, the other thing I meant to say that I skipped over is that bleach is actually effective against spores, but you have to use a much higher concentration and you have to leave it on for a fairly long time. So anytime that you have to leave a chemical on for a long time, that's a sign that you probably don't want to use it as a disinfectant. Remember, these chemicals are going to have effects on materials, some materials, not all materials. And if you have to leave bleach in contact with a surface for, say, 10 minutes, there are some surfaces that are just not going to survive that, right? They're just going to be destroyed by the bleach. So anytime you see a chemical that needs an extended exposure time, that's a sign that that's not a good choice. It's probably not a good choice as a disinfectant. Bleach is really best on like stainless steel, because stainless steel is just really resistant, very, very resistant to destruction. Now, the other place that bleach gets used a lot is with fabrics. We all know, if you've used bleach in the laundry, you know that it has a nice effect. It gets dirt out really well, and it helps white fabrics stay very white. But you probably also know that you can destroy fabric with bleach, right? If you use too high a concentration, or if you leave it on for too long of a period of time, you can burn a hole through fabric with bleach. So you really have to know the surfaces that you're going to be trying to disinfect when you try to choose a good chemical. Now, on this slide, we're looking at the limitations of chlorinated compounds, and specifically this most common one, which is bleach. Down at the bottom here, you can see a Clorox product in a spray bottle. There are so many bleach-based cleaning products on the market. It's crazy, but people like bleach. People feel like bleach is going to really clean and disinfect their home. So they're very, very common. Most bleach products, though, that you're buying are not just bleach. They have other molecules added into them as well. So here's the list of limitations for bleach. Bleach does not work nearly as well in any environments, any surfaces that have a high pH associated with them. Remember, we think the bleach is effective because it acts like a weak acid. If you mixed that weak acid with a basic environment, you're going to neutralize it, and it's not going to be as effective. So less effective in a high pH environment, less effective in the presence of organic matter. Again, think dirt, soil, blood, vomit. It's just going to be less effective in the presence of any organic material. So you always have to clean first and then use your bleach. Something that is also really important in a practical sense is that you have to change bleach solutions frequently. When you mix up a bottle of 10% bleach to use as a disinfectant, you have to make it up fresh about once a week. So yes, bleach is a lot less expensive than alcohol is, but you have to throw it away once a week and make up a fresh bottle of it. So it's something to think about. Bleach absolutely damages certain materials. It will corrode metal over time. So you have to, again, the metal that you're applying it to, you have to know whether or not corrosion is an issue with that metal. And if corrosion can be a problem with that metal, you probably don't want to use bleach on it. It will discolor fabrics. It will take dye out of fabrics. And if you put it on fabric at too high a concentration, it will destroy the fabric. Here's one that you also might be familiar with. If you mix chlorine bleach with ammonia, it creates a toxic gas. It creates chloramine. Every year, there are stories in the news, well, 1 or 2 stories every year about someone being rushed to the hospital because they mixed those 2 chemicals together in their home. You know, it's kind of old school to use ammonia. There aren't a lot of people that use ammonia routinely to clean, but I know several people who use it. If you mix bleach with your ammonia, if you let those 2 chemicals mix together, it's going to generate a toxic gas. You will pass out, and if nobody gets to you, you will die. So you never mix those 2 chemicals together. Gosh, my grandmother used to use ammonia all the time. She cleaned her kitchen with it. She cleaned her bathroom with it. And she also used bleach. She used bleach in the laundry. And it used to scare scare me to death, because I'd be like, Graham, be sure that you never mix those 2 chemicals in a bucket, okay? Keep them separate from each other. So bleach is actually much less expensive than alcohols. So it can be a good option if cost is a real issue. But again, a lot of the commercial products made for residences aren't even pure bleach. They're mixed with all kinds of other things. All right, the next family, the third family on the list, are the Iota Fours. Now, Iota Fours are not the same as iodines. Those are 2 different chemicals. So iodine is the pure chemical, and an iodophore mixes the iodine with what's called a solubilizing agent, something akin to detergent or soap. So this is a bottle of povidone iodine, which is an iodophore. It's got that solubilizing agent in it. And if you've ever used this povidone iodine, one of the common brands is Betadine. You may have heard of Betadine. This is a compound that is used both as a disinfectant and an antiseptic. So it is falling out of favor just a little bit for some newer compounds, but betadine used to be the go-to for surgical prep. So you would scrub the area that was going to be operated on in betadine first. A lot of hospitals still use betadine, but like I said, there are some other compounds that are becoming more popular. Betadine has, or I should say, Iota Fours, because there are others besides betadine, has a bactericidal action against bacteria. So just like alcohol, just like bleach, but it doesn't act quite as fast as alcohol and bleach do. For scope of activity, it is effective against the TB organism. It is effective against fungi, but you have to leave it on for several minutes. It's effective against viruses. It's effective against spores, but only if you leave it on for a long time. Now, what iodophores are doing to bacterial cells is twofold. They denature protein, just like alcohol does, and they also damage DNA. So the reason that they can damage DNA is because they can penetrate through the cell wall. Remember Graham's iodine, right? We said Graham's iodine helps crystal violet penetrate the cell wall. It's the Graham's iodine that's pulling the crystal violet through. Alcohols don't penetrate the cell wall as well as iodophores do. They can't get into the deeper parts of the bacteria as well as iodophores can. So the iodophores have a little bit of a secondary effect by destroying the DNA as well as the protein. Now, limitations of use for iodophores. Here's probably the strangest one right here. There are some reported species of bacteria, not very many, but there have been a couple species reported that can actually survive in iodophore solution. So this is very disturbing. Now, I can't say that there are pathogens that can survive in it because I don't know of a single pathogen that can survive. But there have been cases in laboratories where people have used iodophore solutions in spray bottles, and they leave the solution in the spray bottle for long periods of time. And when they go to check, there's bacteria growing in it. So very, very scary. It's a reminder that you want to change your solution frequently if you're going to be using this compound. Just like with the bleach, you want to change it up frequently. Now, iodophores are nice in terms of damage. They're much less damaging to materials than alcohol and bleach are, but there's one material that they will destroy, and that's silicone tubing. So if there's anything like in a hospital setting or in a doctor's office made out of silicone, and there are lots of medical supplies made out of silicone, you would never want to routinely disinfect it with iodophores because they'll eat it away and destroy it over time. So what the iodophores are able to do that some other disinfectants are not able to do is what we're looking at on this slide. What you're looking at right here represents a piece of membrane that was taken off of a bacterial cell. So they're showing us the phospholipids. You can see the round heads on each side of the membrane and the tails in the middle. So this is a piece of membrane. And these structures, these represent proteins in the membrane. Yeah. This represents the inside of the bacterial cell. And this is the outside. So you can see the DNA is in here. The DNA molecule is in here. There are some proteins inside the cell as well. Now out here, they're showing us these two molecules right here. This is the iodophore. So they've labeled it as PVP iodine. This is the iodophore combination. And what happens when you use it as a disinfectant is the iodine sort of comes out of it, dissociates from it, and penetrates through the membrane. It'll destroy the DNA and it will destroy proteins. It'll unfold proteins and it'll kill the cell. Now, there's one more family on our list, and these are called Quaternary ammonium compounds. Very strange name. You may or may not have seen this chemical in the doctor's office or in a facility where you might work. Hibiclens. Hibiclens is a brand name for a compound called chlorhexidine. And chlorhexidine is a very popular quaternary ammonium compound. So what these molecules or chemicals are made out of is an ammonium ion attached to what's called a modifying agent. It's the ammonium that's really important on these chemical compounds. There are lots of these on the market right now. There's one called Rocal, there's one called Virex. Again, chlorhexidine, you can buy just brand names of chlorhexidine like Hibiclens, or you can buy generic formulations of chlorhexidine. There's lots of these on the market. They're very popular. They are antibacterial and they are bactericidal, just like all the other ones we've talked about today. They are not as rapidly bactericidal as alcohol and bleach are. Alcohol and bleach have the most rapid rate of bacterial cell death when they're applied. Here's our scope of activity. Quaternary ammoniums are not effective against Mycobacterium, that organism that causes TB. So again, if you were working in a facility where TB was a problem, if you were working in a place that treated a lot of patients who have TB, you would probably not choose this as your routine disinfectant. Effective against fungi, effective against viruses, but here's an interesting note. These quaternary ammonium compounds are only effective against enveloped viruses. Now, the reason they're still very popular is because if you recall from lecture, most animal viruses are enveloped. Most of the animal viruses, and of course humans are just animals, most animal viruses are enveloped. So quaternary ammoniums are antiviral against the viruses that affect us. So that's why we use them. The kinds of viruses that don't have an envelope around them are often plant viruses. So, maybe if you were working in a greenhouse setting, or you were working in some kind of a facility where you were growing lots and lots of plants in the facility, and you needed like some kind of commercial landscaping facility, maybe, you might not want to use a quaternary ammonium against viruses in the environment because it just wouldn't be that effective. They work on enveloped viruses only. Mechanism of action for quaternary ammoniums. They denature protein. We've seen that a few times. And they too have a secondary effect. And the secondary effect is on the membrane. They are going to have an impact on these phospholipids, and that will disrupt the membrane. It will cause the membrane to fall apart. So again, it makes sense that they're effective against enveloped viruses because they have a piece of membrane around them. That's what they're showing us on this slide. There's a drawing right here to represent a bacterial cell. And they've put this little square right here over part of the membrane, and then they blew that up for us. And they're showing us that as we apply the quaternary ammonium, the membrane is going to be affected. It's going to just start to literally fall apart. The ammonium, the NH4 plus that's in quaternary ammonium, that's what these represent. And they're going to interact with the phospholipids and cause them to disassociate from each other. And that's going to just tear the membrane up and kill the cell. Now, limitations of use for quaternary ammonium. They are less effective in hard water. So if you live in a part of the country where hard water is a problem, you have to be aware of that. You can still use them, but they are less effective. The hard water, it just has more mineral in it than other water does, and it actually binds up some of the ammonium, so it's not as effective. Here's one that I find interesting. Quaternary ammoniums are less effective as antiseptics if you apply them with cotton. So if you think about the operating room, if you were using something like Hibiclens to clean off a surgical site, you would not want to apply it with cotton gauze because cotton binds up the ammonium and it's just not as effective. So you might want to try a sponge material that was not made out of cotton because cotton will literally interfere with the quaternary ammonium. Just a strange little fact about these chemicals. There's another important limitation to the quaternary ammoniums, and it's one that's getting more attention as these compounds get more and more popular in hospitals and long-term care facilities. The people who are applying these, so the janitorial staff, can become sensitized to them over time. And what I mean by that is they can develop an allergy to them over time. Now, this is not a true allergy. It's not that they are allergic to the molecules, but instead, their airways are just becoming very reactive to the fumes from these chemicals. And that's what they're trying to show us here in this picture. This represents a set of lungs. You can see they've drawn in the bronchus here, and they're showing us that the airways in a normal lung are very open, and the airways in a sensitized lung are very swollen and inflamed. So it's the fumes from the concentrated form of the quaternary ammonium that can create a sensitivity over time. And what'll happen is the staff, I've actually met people who this has happened to. The staff will report, oh, I have some tightness in my chest all the time. I'm coughing all the time. Their airways have just become very sensitive, very reactive. And when they get a face full of the fumes from the Virex, their airways literally swell. And it's similar to an asthma attack. So it's not an infection or anything, and it's technically not an allergy. They're not allergic to a molecule, but it's more of a sensitization that happens. So what that means is, for hospital facilities that are using these quaternary ammonium compounds every day to clean, it's important that they provide the janitorial staff with facial protection, with respirators. It's becoming more and more clear that that's important. Now, not every person will become sensitized, but we can't predict who will and who won't. So it's just good practice now to provide your janitorial staff with some respiratory protection if they're mixing up quaternary ammoniums for cleaning purposes. I have used, so when I was in practice, I started off, when I first started off, and I was doing surgery, I used to use mostly povidone iodine. So, betadine. Never had a problem with it. By the time I left practice, there were some places that were using things more like Hibiclens. I know for myself as a patient, when I have had surgery, I've seen both things used. Not too long ago, I had to have an elective surgery done, and the hospital asked me the morning of my surgery, to bathe, take a shower, and use Hibiclens to clean my whole body with Hibiclens. And then when I got into the operating room, I asked, because of course, I'm a scientist, I asked what they would use to clean the surgical site, and they said they would use the povidone iodine. So they were essentially using both to try to have an antiseptic effect. So you really do. You see both of them used in hospital situations. Okay. I'm just trying to read through to see if what kind of questions I can answer in the couple minutes we have left. Do I think most penicillins have already been found? Yes. Anything that's a natural penicillin type drug, probably already been found. But there's still lots of opportunities to modify penicillins in the laboratory and make these semi-synthetic antibiotics. Lots of opportunities for that. But in addition, there are companies that are studying brand new types of antibiotics. There are people who are searching for natural antibiotics, searching the world for natural antibiotics, and there are people who are trying to build better antibiotics in the lab. So don't lose hope on antibiotics. It's just that we need to prioritize finding them and making them. We need to prioritize that so that drug companies, have an incentive to keep looking for them. The other question I see is about whether or not other countries can still add antibiotics. Oh, yes. Yeah. Now, a lot of the countries in the European Union, very strict about antibiotics in animal food. In fact, they're more strict than we are. They've always been more strict than we are. So any country that's in the European Union has a very safe meat supply in terms of antibiotic residues in the meat. But when you leave the European Union and you go into some of these other countries, antibiotics are still a real problem. We know that in Russia and some of the countries around Russia, it's a big issue, feeding antibiotics to animals, and also in China. China has very few regulations around what can and can't be fed to food animals. That's a great question. And again, we don't feed the animals a level of antibiotic when it's done that is high enough to kill bacteria. So that means animal feeding of antibiotic does not contribute to resistance. I know that seems strange, but the only way that antibiotics cause resistance is by killing off some bacteria and leaving other bacteria. So actually, the feeding practice was not responsible for creating resistance, but just the use of antibiotics therapeutically can lead to resistance. But you and I, we want to eat food, at least I know I do, that isn't adulterated. If I'm going to eat chicken, I don't want the meat to have drugs and chemicals in it. And the good news, again, for us in this country is that it's not a problem here. But in other countries, it can absolutely still be a problem. They just don't test for it in other countries. They don't look for it. And so the way we know that it's a problem is when they come in as imports. There's a certain sampling that's done, and we test for antibiotics. And a lot of the stuff, for example, that comes in from China has high levels of antibiotic in it. So we're very lucky. We actually have a very safe food supply here in our country. unadulterated food supply. Okay, so the other one that I think it's Vicky who's asking this. I don't have my reading glasses on. So the question about the antibiotics before you go to the dentist, it's one of my favorite questions. Remember when we talk about bacteria in the human body, when we talk about our microbiome, those bacteria and other microbes are attached to surfaces. So the microbiome on your skin, you can't wash it off. The microbiome on your teeth and in your intestine, it's essentially adhered to the tissue that it lays on. But there are what we call planktonic bacteria that are not attached, and they're not part of the microbiome. Pathogens are planktonic. They sort of move around individually and cause disease. So planktonic bacteria, if they find a place in the body where they can attach and grow as a film, they will. And one of the places that that can be a real problem is heart valves, artificial heart valves, and heart valves that are damaged and aren't functioning correctly. So what we've learned over decades of time is that when we go to the dentist and they clean our teeth, and you've all experienced this, the cleaning process makes your mouth bleed. It makes your gums bleed a little bit. And planktonic bacteria, again, free living bacteria, can get into your bloodstream while you're getting your teeth cleaned. Most people don't have any problem with that because you have a functioning immune system, and your immune system is going to catch those bacteria and going to destroy them, and you won't even notice. But people who have heart valve problems, those planktonic bacteria can land on those heart valves and can grow. And we've only learned this through tragedy. Again, people who have sick valves that are not functioning correctly, the bacteria from their mouth gets into their blood. It will land in the heart valve, on the heart valve, and it will grow, and it can cause very severe, fatal endocarditis. It can cause heart valve infection. So what we've learned over time is any patient who has valve disease should be given an antibiotic before they go to the dentist. They get an antibiotic before they go. So the antibiotic is circulating in their blood. And then when they get their teeth cleaned and their gums bleed a little, and that planktonic bacteria gets into their bloodstream, there's already antibiotic in there, and it will kill the bacteria. Oh, there have been just tragic stories about people who didn't know that they were supposed to do this. And they just go in for a routine teeth cleaning. And 2 weeks later, they're dying of endocarditis. Very, very sad. But almost any dentist now knows to ask, do you have any heart disease that we should know of? Because if you do, that you're going to get some antibiotic before you come in for your teeth cleaning. Great question. Anything else? I've kept you over. I need to let you go. But great discussion today. And I'm always happy to talk about anything we're talking about lecture while we're together for these labs. They're such, these topics touch on a lot of other things, right? I mean, the topic of antibiotics and antibiotic resistance is a really important, timely topic. So feel free anytime. All right, I'm going to let you go. Have a great evening. And if you need anything, let me know. Send me a message. And we will see you next time.
Thank you.